Hello students, this is Dr. Faizan Mirza. Here in this video, I'm discussing with you the paper of Feb March 2024 and the MCQ paper here of AS level. So let's start with this. The first question that came here dealt with the microscopy. As we know that the questions follow the order which is given in the syllabus. So the first few questions are actually for the first part of the chapters or the first part of the syllabus. And as the syllabus progresses, so the questions also follow the same trajectory. So question one is a student, they are saying the student uses light microscope to observe a blood smear on a microscope slide. An eyepiece graticule was used to measure the diameter of a white blood cell on the slide. The student recorded the white blood cell was 5 eyepiece graticule units diameter. So this is what's provided to you, that this was the measurement that the student took. What additional information does the student need to determine the diameter of the white blood cells in micrometers? So we know that the student already is using eyepiece graticule. So what the student should do? The student should use a micrometer, and using a micrometer, the eyepiece graticule should be calibrated, and using that calibration, this is enough to calculate the actual diameter of that white blood cell. Nothing else is required. The magnification is of eyepiece is not relevant here, so that cancels out both the options B and C. Again, the magnification of eyepiece and the magnification objective, this is not of any concern since the student is just using the eyepiece graticule, and the eyepiece graticule need to be calibrated using the same magnification at which it was basically measured. Question 2. Which statement explain why it is necessary to use electron microscope to see the crystals of a mitochondrion? We know the mitochondria and crystals are very tiny structures. So yes, magnification is relevant, but more than magnification, resolution is more relevant here because resolution allow a much detailed image to form of structures which are very tiny. So magnification electron microscope is greater than light microscope. Yes, the statement is correct, but not relevant for this answer. Similarly, the membranes of crystals are separated by distance greater than 200. No, not greater than 200. The wavelength of electron beam is longer, and then the visible wavelength. This is option D. This is not true. The maximum resolution of a microscope using visible light is too low. Yes, we know that visible light resolution is low as compared to that of electron microscope. That's why we are able to see objects which are actually very small in structures. Some stain can be used to identify the cell structures in living cells. The Dell solution of one stain can cause this cell structure to appear blue. The blue color rapidly disappears from most cell structure except for the ones that release energy. They stay blue. Which structures are likely to stay blue? So energy we know in the form of ATP is being produced in the cells either from the cytoplasm or from the chloroplast or from the mitochondria. So from the available information, only option D is correct. When nucleus secrete from goblet cell, these are the events that take place. These are the various steps mentioned. You just arrange them in the order. We know that the addition of carbohydrate is the first step, followed by the separation of vesicle from the Golgi vesicle. This is followed by the fusion of the vesicle from the cell surface membrane. This is part third, and then the exocytosis of the glycoprotein. This should be the fourth. So the only option that stands correct is this. 4 and 4 both cannot be the first step, and obviously 3 and 2 cannot. This is just two cannot be the last stage. Yes, 3 can be, but 4 cannot be the first. So C, D, and B are not the options. The diagram shows for biological molecules, which biological feature are present in the typical prokaryotes. So DNA, RNA, 70 ribosomes are all there. We know capsid are protein, they are present in virus. So we are dealing with prokaryotes here. So the answer is anything but 2. Which row shows the features occur in dicot plants and in typical bacterial cells? So both must qualify for the answer. 70S ribosomes are present in both. 80S ribosomes are present in one but not the other. So yes, centrioles are present in both. No, they are not. So this is wrong here. B is the only option which says 70S ribosome is there. 80S ribosome is uncommon in both. Central is common in both. No, it's uncommon. Circular DNA is common to both. Yes, so option B is the only correct option. Similarly, in the other options, you can also figure out which option cannot qualify for the overlap between both cells, which is a dicot plant and a typical bacteria. Which flowchart using outlining of the cell of test for not using sugar? When you are testing for not using sugar, do you add alkali first? No, we don't. So option A and B are incorrect. We boil it with HCl. Yes, option C and D both seems to be right. We neutralize them with sodium hydrogen carbonate. Yes. And then we add the Benedict's in boil. We do not add Biuret. Biuret is for protein test. So option C is correct. Which diagram show the release of water molecule during the formation of a glycosidic bond? So if you look at these structures here, these are all beta glucoses. So beta glucoses and water molecule is being released. So yes, all the three can form glycosidic bonds. Which molecules have a structural formula that contains C double bond O? That contains C double bond O. So glycerol, if you can recall glycerol molecule, a glycerol molecule is having three carbons, and all carbons have OHs attached to them, and the remaining bonds are filled with hydrogen. So there is no C double bond O here. So glycerol is not an option. Proteins have carboxyl group, fatty acids have carboxyl group, amino acids have carboxyl group. So the answer is 1, 2, and 4, which is B. Which statement about triglyceride is correct? Triglyceride, we know there is one glycerol molecule attached to three fatty acid tails, which means three ester bonds. So in any triglyceride, all fatty acids are saturated? No. All unsaturated? No. There is no such rule. They may be unsaturated, saturated either. Any combination of these. Two triglycerides? No, not two, three. So that's incorrect. Triglycerides are polar? No, they are not polar. Hydrophobic, polar and hydrophobic. Polar hydrophobic, there's nothing like polar hydrophobic. They are polar as well as hydrophobic. Again, polarity is not there because triglycerides are non-polar. Glycerol is joined to each fatty acid by a covalent bond. Yes, that's a covalent bond that attaches them to the glycerol chain. So 10, the answer is D. Question 11, many flowers produce a sweet solution called nectar. Bees provided a solution, the nectar use enzyme Q to change the nectar into honey. So here we are given information that there is something called nectar. The nectar is converted to honey using enzyme Q. In this case, enzyme Q is the enzyme, nectar is the substrate, and honey is the product. So keeping these in mind, after testing the sample of nectar for the presence of reducing sugar, using standard laboratory reagents, the sample was blue, which means they lacked reducing sugar, which means the nectar is the substrate, it lacked reducing sugar. After testing the sample of honey, which is the product, in the same way, the sample was orange, which means reducing sugar was present. What conclusion can be drawn about a reaction catalyzed by enzyme Q consistent with the result? So substrate is straight blue, it must be sucrose, it's a non-reducing sugar, and product should be a reducing sugar, could be either one. So all options are correct, but as far as the substrate is concerned, only option B and C seem to be correct. So we actually narrow it down to option B and C. Now we look at the reaction. We know that the type of reaction which involves enzyme is hydrolysis and not condensation. So we omit out B and we are left with the option which is C. The diagram shows the structure of a part of a peptide to glycan molecule. So here you can see that this is the carbohydrate. This is beta glucose, which is flipped. You can see the flipping here, and this must be a beta glycosidic bond. Then here you can see the chain of amino acids. This is 1, 2, 3, 4. Some 5 amino acids are there, and they're asking what type of 1,4-glycosidic, but which type of 1,4-glycosidic linkage are there, and how many peptide bonds are shown in the molecule. So the type of glycos this linkage is beta 1,4. Why beta 1,4? Because we know that the molecule is flipped. So yes, it must be beta. As far as the number of peptide bonds are concerned, so you can just count the number of peptide bonds. What I have done, I have encircled the amino acids here. So you can just count the number of the peptide bonds here. So it comes out to be 1, 2, 3, and 4. Whenever you are trying to figure out which amino acid is there, what do? We just try to figure out the central carbon. A central carbon of an amino acid is one which is always having hydrogen on one end, an R group on the other end, an amine group on one end, and a carboxyl group on the other end. So using this, we figured out all the amino acids here. And here the amino acid is being changed slightly. So yes, this must be an amine. This should be an amine group as well that got changed because of addition of this reaction here that occurred by attaching or by bonding the carbohydrate with the amine terminal of the central carbon of this amino acid. So this is something which you can this regarded as AO2 assessment objective 2, because it's I think it would be a common thing here for the student to pick out the answer of beta 1,4 and the peptide bond is 3, whereas the peptide bond here are 4. A scientist investigated the progress of two enzyme catalyzed reaction in separate test tubes X and Y. So X and Y are both two separate test tubes. Both reactions in the results in color changes that can be detected using colorimetry. 0.5 cm cube samples were taken from each test tube at the start of the investigation at regular intervals for the next 5 minutes. So for 5 minutes, you can see that the trajectory of both test tubes, X and Y, shown to you with the percentage absorbance. Copper ions were added to each sample as soon as the sample was collected and activate the enzyme. So copper ions here act as an inhibitor to stop the reactions to stop the reaction from progressing further. The absorbance of each sample was measured using a colorimeter. The graph shows the results of this investigation. Which statement is the consistent with the results of the show, which is shown in the graph. So if you look at the y-axis percentage absorbance, on the x-axis time in minutes. So as the reaction progresses, we see that test tube Y contents the substrate in Y, which must be at the start of the reaction, turns into product in Y. So we see that the substrate Y had a substrate in the test tube Y had a high absorbance. The product of Y is having low absorbance. Similarly, in test tube X, the substrate initially, which is at time 0, must be substrate of this test tube. It is having a low absorbance, but as the time progresses and it converted to product, so the product is having a higher absorbance. Which statement is consistent with the results shown in the graph. So that's the only option which stands correct. If you annotate this graph properly, it's not difficult to figure it out what they actually are asking here. This MCQ is not about the graph of product and reactant. Both X and Y are two separate test tubes. So product in Y, test tube Y has a lower absorbance. So yes, test tube Y, the product is having a lower absorbance and the substrate is having a higher absorbance. So yes, option B is correct. Rates are not displayed. You cannot calculate the rate here. The substrate in tube X, this is completely reversed, so that's incorrect as well. A student investigated the hydrolysis of lipid in high-fat milk using lipase enzyme. So one thing to keep in mind is whenever lipase is acting on lipids, it always results in formation of fatty acids. And fatty acids are cases they will reduce the pH. They will make the pH more acidic. The temperature was kept constant. pH of reaction mixture was recorded at 0 minutes to at every interval for 20 minutes. So the pH must keep on decreasing. As the fatty acid will increase, the pH will keep on decreasing and becoming more and more acidic. Which statement correctly describes the expected results of this investigation? The product forms more slowly as the time proceeds because the concentration of substrate is decreasing. Yes, because as the substrate becomes lesser and lesser, the rate of reaction slows down. The pH of the reaction mixture increases. No, it decreases. The increase in the concentration of the product eventually cause the lipase molecules to denature. Yes, because when the pH will change, it will become acidic. It means that the pH changes, so the molecules of the enzymes will become denatured. So yes, 1 and 3 correct. The correct measurement of the bit of cell membrane, we know we have learned this value. It's about 7 nanometers. So the only answer that qualifies in the range is B. Where in the cell surface membrane are carbohydrate chains of glycoprotein and glycolipids mainly located? Now we know that glycoprotein and glycolipids are present on both sides of the membrane, but where are they mainly located? Mainly they are located on both on the outer surface. Mainly. So yeah. Sodium ions can enter the cell across the cell surface membrane. Sodium ions are small, they are polar. Which method could be used? Active transport only? No. Active transport is used? Yes. Passive diffusion can also be used because there are channels that will allow sodium ions to move. For example, in neurons and muscles, we have channels that open up. There are leak channels that allow sodium to move in or move out depending on the gradient. And yes, we have the sodium potassium pump as well. So all the ions, sodium, potassium, protons, chloride ion, they move by active transport as well as by passive diffusion. The diagram shows the artificial partially permeable membrane was used to separate 5% sodium chloride with 10% sodium chloride. 5% sodium chloride we know is dilute solution, 10% sodium chloride we know is a concentrated solution. A concentrated solution is always having a lower water potential, a dilute solution is always having a higher water potential. So whenever the concentrations are mentioned to you, I recommend my students to just annotate this together with the diagram and predict the direction of osmosis yourself. Because in osmosis, water moves from a higher water potential to a low water potential. In short, it moves from a dilute solution to a concentrated solution. In other words, it moves from 5% sodium chloride to 10% sodium chloride solution. The volume in R should decrease, volume in S should increase. So volume in S increases, net movement of water from high water potential to low water potential. So straight away, the option A is correct. No other option could actually qualify for because they all are incorrect signs. So yeah, option A. That's it. Question 19. Agar cubes can be used to demonstrate the effect of diffusion on changing the rate surface of surface area to volume ratio. Three different Agar cubes were made using colored indicator solution were placed in dilute acid that diffused into the cubes. Okay. As the acid diffused into the cubes, the color of the indicator solution changed, which means the color inside this cube will start to change. The cubes had volume of 1, 2, and 3 cm cube, but left in the dilute for the same time. All the variables were kept constant. After 10 minutes, the Agar cubes were removed. They were cut in half. The cut surface were observed. For the results are recorded in the diagrams. All diagrams were drawn to the same scale. The results of 2 cm cube was shown. So this is the 2 cm cube Agar cube, and this is the original color that you can see inside. And so as the acid would have moved in, this color would keep on decreasing. So why it would keep on decreasing? Because it would keep on changing. The color changes because the acid comes in. So this is the diagram shown for the 2 cm cube, which diagram show the results of 1 cm cube and 3 cm cube. So we know if 2 cm cube is having this much color left of original color, so 3 cm cube, which is the larger cube, should have a bit more of the original color left because it will allow slower exchange. Whereas a cube, which is much smaller, it will have lesser quantity of the original color left because it will allow a quicker exchange. Yes, the time is not long enough for all the color to disappear. So option A and B are incorrect. Option C and D both show 1 cm cube as the same. So option C and D both qualify for the answer as far as the 1 cm cube is concerned. But when we look at the larger cube, the first option C shows the correct order, but option D shows a smaller area still having the original color, whereas the smaller square having a larger area. So that's not possible because a larger cube allows slower exchange. So a slower exchange, it will take more time for the dilute acid to enter and change its color. So option C is correct and no other option. During sperm formation in mammals, part of structure of each chromosome is replaced with proteins called protamines. This replacement allows DNA to be packaged much more densely than would otherwise be possible. For packaging of DNA, we need histone proteins. Which part of chromosome is replaced by protamines? So we know histones are the only ones responsible for packaging, so straight away option C. The diagram shows the mitotic cell cycle, during which phase of do chromosomes condense and become visible. So this A, B, and C shows interphase, G1, F, and G2. We are told this is cytokinesis. So D must be the M phase. We know that the chromosome condense in early prophase. So option is D. Question 22. Which statements correctly describe features of stem cell that are essential for their role in cell replacement and tissue repair? So we are talking about two things here, cell replacement as well as tissue repair. So tissue repair means that the stem cell must be residing within the tissue. That's why it gets repaired. And cell replacement, yes, it will need division to occur so that a new specialized cell can form. Both must be there in the option for 1, 2, 3, or 4. After mitosis, stem cell daughter cells either remain as stem cells or follow a developmental pathway to form specialized cells. Yes, that's required because we know that in every tissue, stem cells are there that can replace the cell and still have the depot of cells left for later on use. The stem cells are different to all body cells because they retain all genetic information in their DNA throughout the life of organism. Within a tissue, this is not possible because every tissue, for example, your bone marrow and your skin cells, they both have stem cells, but the stem cells are not exactly alike anymore, especially when we are talking about the person who is not a zygote anymore or not in the fetal life. A small population of stem cells retained in the body adults throughout their lifetime. Yes, there is a small population left. Stem cells have more telomeres than the body cells. This allows them to undergo a limited number of mitotic divisions. This can qualify for replacement, but not for tissue repair. It's not a relevant option as far as both the tissue replacement, cell replacement, tissue repair is concerned. So we are left with the option 1 and 3. So if you omit out 2, even if you omit out 2, you're confused with 4. Even if you omit out 2 because this is outright wrong, so this nullifies all the 3 statements. So the only good answer left is C. Which statements are all correct for all nucleotides? Nitrogen containing base is always attached to first carbon. Yes. Phosphate group is always attached to carbon fifth. Yes. A condensation reaction occurs to join nitrogen containing base to pentose. Yes, that's true as well. Just recall this. Recall what nucleotide was. So all three are correct facts here. Nucleotides are linked together by condensation reaction between phosphate groups. No, a phosphate group of one is attached to the carbohydrate, this carbon of the next nucleotide, not the phosphate. That's how the 5 to 3 carb bonds actually form, 5 prime to 3 prime. They are not exactly the bonds between phosphates. So yes, as far as the options are concerned, 1, 2, and 3 are correct. Fourth is wrong. How many phosphodiester bonds are present in a circular DNA molecule having 2700 base pairs? So you just multiply that by 2. The number of base pairs provided to you, just multiply it by 2. You will get the total number of phosphodiester bonds. So 27 by 2 gives you 400. Since it's a circular DNA molecule, that's why we are doing this. If it was a linear DNA, we would subtract 2 from here. So we would reduce it. The answer would have been 5398 if it was a linear DNA. But since it's a circular DNA, the answer will be 5400. Which statement about complementary base pairing are correct? We know purine pyrimidine of different sizes. Complementary base pairing occur during translation. Yes. Base pairs are of different lengths. No, they are not of different length. All purine pyrimidine base pairs have the same length. Uracil form two hydrogen bond with adenine. Yes, because only guanine and cytosine have these triple bonds. So 1, 2, and 4 are correct. 3 is wrong. So again, you can use the same thing. If you were able to highlight that the third option was incorrect, you nullify all the three options A, C, and D. You are left with only option B. Question 26. During a mitotic cell cycle, the chromosomal DNA is replicated. The specific points on the DNA molecule where replication occurring is called our replication forks. This is something new for the students. We don't really discuss replication forks at this level, not the demand syllabus. So that's why they have given you information here. A typical human chromosome has about 150 million base pairs of DNA. Okay. It takes about 1 hour to replicate DNA of a typical human chromosome. 1 hour means 3600 seconds. The rate of replication using a single replication fork is 50 base pairs per second. So one replication fork, 50 base pair per second. Three facts are provided here. Approximate. How many replication fork must be there for a typical human chromosome during replication? So what I've done here is I've just worked it out that one replication fork is actually 50 based on 50 base pairs. So for 15 million, you will have X. So just cross multiply. I got the answer 300,000. And since this was 300,000 was for per second, so I need to just for per hour, I'll calculate by dividing by 3600, which means for 1 hour, my answer came out to be 834. 834 is closest to 835 among all the options, so that's the answer. Which molecule has its synthesis directly controlled by DNA? So that's protein. Amylase is an enzyme of protein, so option A is correct. Question 28. Which statement correctly describes the association between a companion cell and seed tube elements? Now, this is a new type of question. The companion cell provide all ATP, an energy process for both cells. Both cells do not use ATP. A companion cells control active transport through the sieve tube. No, they don't. Active transport is only between the companion cell and the source cell. Companion cell prevents side-to-side movement of assembly between the sieve tube elements. No, again, this is by plasmodesma. The companion cells provide a nucleus. Yes, that controls the cellular activities in both type of cells. Because all three options are incorrect, so yes, option D is the only one that we can actually narrow it down. And we know that these sieve tubes do not have companion cells. Oh, sorry, they do not have a nucleus. So the nucleus of the companion cell is actually working for both. Question 29. A maize seedling will grow in the soil containing lanthanum ions labeled with a chemical that fluoresces under ultraviolet light. The diagram represents what was observed when a fruit was examined using light microscope with ultraviolet illumination. This is xylem, this is Casparian strip. If we know, we can see fluorescence, you can see in the root hair cells, fluorescence is there, and the cortex cells, the fluorescence cells, fluorescence is there as well. But at the Casparian strip, the fluorescence does not proceed. So it means we know that Casparian strip blocks the apoplastic pathway. So apoplastic pathway is the one which is relevant here. Symplastic is not occurring. And beyond Casparian strip, apoplastic is not occurring. What is correct conclusion about the transport of the lanthanum ions in the maize roots? The ions are not transported through apoplastic pathway. No, they are transported by apoplastic. In fact, symplastic is not used. So symplastic is not being used. We can cancel out C, D as well as A. That is transported by apoplastic pathway only. Yes, B is correct. Which description of adhesion and cohesion is correct? Just recall the facts. Adhesion is between water and the wall, and cohesion is between water molecules. Option D straight away is correct. Question 31. Which conditions are needed to allow the mass flow of sucrose and Phloem C2 elements? Phloem C2 elements and sucrose. High hydrostatic pressure is required, we know that. But a lower water potential, because when the water potential is low, only then more and more water can enter and more hydrostatic pressure can actually be produced. Because if it was a higher hydrostatic pressure, the water will stop moving inside. So yes, both actually the first column in both includes high hydrostatic pressure, but high water potential is not correct. Low water potential is correct as far as the C tube is concerned. But when we move to sinks, in sinks we know that a lower hydrostatic pressure because the quantity of the substances decrease down because they are moving to the sink, and the water potential stays high because then the water starts to move into the surrounding cells. Question 32. One type of congenital heart defect is where the left and right atria are not completely separated. This is called atrial septal defect, ASD. ASD usually results in blood moving from left atrium to the right atrium. We know that's not the right pathway. This causes increased blood pressure in the right atrium, decreased blood pressure in the left atrium, which will describe the other effects of ASD. Now we know if there's a higher pressure in the right atria, which is told to us, this will increase the pressure in the right ventricle, which in turn will increase the pressure in the pulmonary artery. When more blood goes to the pulmonary artery, more blood goes to the lungs, better oxygenation will occur. Similarly, we are told that the pressure in the left atria drops down, which means that the pressure in the left ventricle will drop down as well, which means that the aorta will have less blood again. So using this information, option D is the only one that stands correct. So that I think can be done if you just recall the pathway that the blood follows. Which property of water is related to its role in blood and tissue fluid is correctly described. So water is solvent for all biological molecules? No, not for fats because they are non-polar. Water is solvent for non-polar? No, it's a solvent for polar. Water requires little energy. No, it needs a lot of energy to increase the temperature. That's why it's having a high heat regulating to vaporization and high specific capacity. Water cools down slowly. Yes, because it's having a high specific capacity. Yes, that's correct. So option D is good. 34. Which statement about tissue fluid formation is correct? The hydrostatic pressure of the blood decreases from the arteriole end and the venual end of the capillary. Yes, because as the blood moves along in the capillary tube, the capillary bed, the hydrostatic pressure starts to drop down. The water pressure always higher in the blood capillaries than the tissue fluid. Always higher is not possible. No cells move out. No, white blood cells do move out. Most tissue fluid formation occurs at the venual end. No, the tissue fluid formation occurs at the arterial end and the reabsorption occurs at the venual end. Oxyhemoglobin, HbO2, carbaminohemoglobin, and hemoglobinic acid and carbonic anhydrase are found inside the red blood cells. How many of these substances will show an overall decrease in the concentration as the red blood cells pass through the capillary? So we know HbO2 will increase because more oxygen will become attached to hemoglobin. Carbon monohemoglobin will decrease because carbon dioxide diffuses out. Hemoglobin also decrease because again carbon dioxide leaves in the form of H plus ion combining with CO3. So CO2 goes out. Carbonic anhydrase is the enzyme. It should stay unaffected. So only two things here decrease. So yes, answer will be 2. Which of these structures typically contain cartilage and cilia? We know bronchi, bronchioles, and trachea. Bronchioles do not have any cartilage. So again, in this question, 1 and 3 are correct. So this option C is what we can go with. Which statements about the function of tissues found in the human gaseous system are correct? Collagen in the bronchi prevent collapsing. No, cartilage is actually doing that. Smooth muscle in the bronchioles can contract. Yes, they can contract to increase the flow. No, to decrease the air. Elastic fiber in the alveoli stretch and recoil during breathing. Yes, they do stretch and recoil during breathing. So in all these options, only 3 is correct. 38. Antibiotic resistant strain. This are major problem when treating TB. A new antibiotic, Texobactin, could be very effective at killing this bacteria. With a small risk of that bacteria will evolve the resistance against this antibiotic. Penicillin and similar antibiotics bind to a single protein. But this new antibiotic binds to 2 lipids that are needed for the formation of the bacterial cell wall. So it binds to these 2 lipids that are involved in bacterial cell wall formation. Then they are saying this antibiotic binds to the region of the two lipids that do not vary across different species. So this is what is shared different in between different species. So this antibiotic can be used for treating infections of various bacteria because they share this target site. Which statements help to explain by the use of this antibiotic is thought to be less likely to lead to evolution of antibiotic resistance than the use of many antibiotics such as penicillin. A single mutation can result in the bacteria that are resistant to penicillin and similar antibiotic, but at least two mutations are required to produce this resistant bacteria. Yes. So a single mutation, the chances are low. Two mutations occurring, the chances are even lower. So yes, one can be correct. Two, mutations can affect the structure of protein but cannot affect the structure of lipids. Yes, lipids do not alter their structures because of mutation, because mutation is in DNA or RNA that changes the structure of protein, which will not affect the shape of lipids. So second option is just not correct. So if two is wrong, that means A, B, and D all are wrong. C is the only option correct. So 1 and 3 are right. Third option, the lack of variation across many species of bacteria and the two lipids that bind to the type. This thing suggests that the particular structure of these lipids is essential for successful bacterial cell wall formation. Yes, it can be used in multiple bacteria. So 1 and 3 are correct. Monoclonal antibodies are now being used to treat some human diseases. What explains the monoclonal antibodies are suitable for this purpose? Monoclonal antibodies can divide. No, they cannot divide. Only cells divide. They are specific to particular antigen. Yes, they can be modified, and they do not act. They can be modified. Yes, and once they have been modified, they do not act as antigens. If they were not modified, they would act as antigens. So option 2 and 3 are correct. So in this way, 39C. 40th. Measles facts are not there in the syllabus anymore. So the question is again of generalized information of what vaccination would do. Measles is an infectious disease caused by a virus. You are given the information. The graph shows the number of cases of measles each in the country before and after vaccine was introduced. So you can see vaccine was introduced. So the cases they stopped. They actually there's a sharp decline and they stayed low. And in fact, the cases disappeared altogether. What could have caused the decrease in the number of cases of measles after vaccination was introduced? Vaccination provided artificial active immunity. Yes, we know vaccination is artificial active immunity. Vaccination provide passive immunity and artificial. No, that's incorrect. Few people are able to act as hosts of the virus. Yes, herd immunity is there. So this is correct. This is wrong. This is correct. So option A is what actually is going to be the answer. So I hope this video was helpful for you in solving this paper. What I would recommend is that you just try to attempt this paper by yourself and see how effective was this video for you. That's it from my side. Thank you so much.